Hello everyone. In this lecture video, I'll be talking about double-strand break model. Sometimes it is also called as a double-strand break repair. Now, this double-strand break repair model, it may basically based on the recombination. But before we go into detail of recombination, let me tell you the definition of recombination. What is it? It is nothing but the breaking and rejoining of two parental DNA molecules to produce the new DNA molecules. It means that it based on the breaking of DNA molecules. Suppose these are two different DNA molecules, then here the cutting takes place over here and then followed by the joining of these two different DNA molecules, which results in formation of DNA which is totally different than than either of the parental cell. This is known as the recombination. Now, in detail about this recombination, see there are different types of recombination. Here I'll focus on the homologous recombination or the general recombination and the site-specific recombination. Homologous recombination is basically it takes place whenever the transformation takes place, and this homologous recombination it takes place in a prokaryotic as well as in eukaryotic. Now I would like to give you some example from in prokaryotic cell, like where this recombination takes place. Number one is during the transformation process where the DNA it enters into the recipient cell. Here, if the homologous DNA enters in the recipient cell, then the homologous recombination takes place. Second way is the transduction. So that can be the generalized transduction. Then third one is conjugation. During a conjugation also the DNA from one bacterial strain enter into the other bacterial strain, isn't it? And being homologous in nature, that leads to the homologous recombination. Whereas in case of eukaryotic, you will find this homologous recombination and during the meiosis division. During meiosis division, what happened? That the pairing of homologous chromosome takes place. Homologous in the sense one which is obtained from the maternal origin and the second which is obtained from the paternal origin. At the time of meiotic division, we call it as a haplodization, where the gamete formation takes place. So during meiosis, what happened? The pairing of homologous chromosome takes place, and whenever the pairing takes place, that time this there is a possibility of crossover and cutting and joining of the DNA takes place, that results into formation of the chromosomes which are totally different than either of the parental cell. Okay, so I would like to tell you how the recombination takes place. You can see this. I have shown here two different DNA molecules. One which is shown in red in color and having the A plus, B plus, C plus gene. Other one which is shown blue in color and having A minus, B minus, C minus, C. The crossover has taken place in between A plus and B plus, and because of crossover, what happened? That the A plus is followed by C. I'll just draw it over here. A plus is followed by B minus and C minus. That is what I have written over here. A plus, B minus and C minus. Whereas the another strand would have the genotype like A minus followed by B plus and C plus. Yes, A minus, B plus and C plus. This is known as the general recombination or the homologous recombination. Now the other one that is called as the site-specific recombination. The site-specific recombination is basically seen in case of what we can say specific transduction process where the lambda bacteriophage get integrated into the E. coli chromosome, and here at and in at, int and genes, these three different genes plays important role. So here they have the very small homologous sequence, and that sequence is known as the POP dash and BOB dash, and this O has got the homologous sequence, and thus this is the way by which this lambda get integrated into the bacterial chromosome. Okay, so these are the examples where we can find the homologous recombination as well as the site specific recombination. Now, let's talk about the double strand break repair model. See, as I said that sometimes it is also called as the double strand break repair model or double strand break model. Okay, so some people use the repair word and some people do not use it. Now, let me tell you the difference between. I mean, is there any difference? So the if you look at the steps by which this recombination takes place, by which the repair mechanism takes place, both process are same. But whenever we focus on the repair mechanism, that time this model is known as the or this model help us to understand how the repair mechanism takes place by means of recombination. And if we look at this model from the recombination point of view, that how the recombination takes place, what is the mechanism, what are the steps by which the recombination takes place in the uh, what in meiosis or during the meiosis, or in case of transformation, transduction and conjugation, that time these are the different steps by which the recombination takes place. Okay, so it means that let us conclude that there is no any difference like if we use the double strand break model, or we can use the title like double strand break repair model. Now, from a repair point of view, let me add few things over here. Whenever the DNA undergo any sort of damage, that can be because of mutagenic agent or maybe because of replication. So it is very important that that damage should get repaired, isn't it? Now, in case of excision repair, what happens that it's as I would like to draw a diagram over here. Suppose these are two strands of DNA, and if the out of this two strand, if the damage takes place over here on one strands, then in case of excision repair mechanism, what happens that this damaged parts can be repaired. This damaged part can be repaired by using the nucleotide sequence which is there on other strand, so that we it means that we can use this other strand as a template, and accordingly the damaged parts can be repaired. Again, what I'm trying to tell you that whenever we talk about this two strands of the DNA, if there is a problem with this strand, then this can be repaired by using other strand as a template. Okay, but but the question is, when the damage takes place on both of the strand, both of this strand, if DNA damage like this way, if this is the damaged part, if like these are two strands of the DNA which are complementary to each other, isn't it? And if the DNA damage on both of this strand, then the question is how to repair this kind of damage. Now the answer is double strand break repair model. Here what happened that the repair mechanism or the repair part, it takes the or it retrieves the sequence from the sister chromosome. Means these are the two strands of the DNA, and a problem is over here. I mean, there is a break on both of these strands, so this break can be repaired by considering the other, that is the sister chromosome. I mean this sister chromosome, these sister chromosomes are used as a template, and this repair and this damage can be repaired. So this is known as the double strand break repair model for the homologous recombination. Okay, now let's focus on the actual this model. What are the different steps in this double-stranded break model? So let us consider these two strands of DNA, 5 prime, and this end would be 3 prime, whereas the complementary strand 3 prime, and this would be 5 prime. Okay. Hmm. Now this is the damage part. I mean you can see that break is there on both strands. So this is known as the double stranded break. You can find that both strands have undergone the break. Okay. Whereas if you look at the sister chromosome, then you will find that both of these sister chromosomes are intact. Next step would be the endonuclease will come in the picture, and it break the uh, it breaks the uh, these two strands. I mean the one by one the nucleotides are removed, and that we this nucleotides are removed in both the direction from 5 prime to 3 prime, as well as from 3 prime to 5 prime, and we call it as a processing of the DNA. Processing of the DNA takes place, which results in formation of uh, 3 prime single stranded tail. I would like to show you where is 3 prime single stranded tail. See, this is nothing but the 5 prime end. I'm talking about the lower strand. Okay, so this is the lower strand, and that is the 5 prime end, and I'm moving towards the 3 prime end. Okay, so you can see that there is extension at 3 prime end, whereas this would be the 5 prime end, and the cutting has processing means what, this much DNA strand has been cut. I mean one by one the nucleotides are removed in this direction, and that results in formation of 3 prime single stranded tail. Okay, now this tail having the 3 prime end, that invades into the sister chromosome. Okay, so this is known as strand invasion by the 3 prime end. Now this will continue to move along with the sister chromosome till it finds the complementary sequence. The moment when it find the complementary sequence, you can see that the synthesis of another strand takes place by means of DNA polymerase. As you know that the condition of DNA polymerase is that it required the 3 prime hydroxyl group. So do you find the 3 prime hydroxyl group? Of course the 3 prime hydroxyl group is free over here, and by using that as a primer, the DNA polymerase will add the one by one deoxyribonucleotide over here, and for that it of course it utilizes this strand as a template. This would be the template. Okay, so by using this template strand, synthesis of this strand takes place. Same thing happen over here. Now this has got the 3 prime end. As I told you that this is the 5 prime end. So obviously this end would be the 3 prime. So again this has got this blue strand as a template, and DNA polymerase will utilize this blue strand as a template, and it will go on adding one by one the complementary nucleotide. And because 3 prime hydroxyl group is available over here as a primer, so the synthesis will take place in this direction, whereas synthesis of this strand will take place in this direction. Okay, and that results into the branch migration. This branch will get migrated in this direction. Okay, so you can see the branch migration in both. I mean this branch will migrate and it will form the intermediate with two holiday junction. Holiday junction, this is known as the holiday junction. Okay, so presence of two holiday junction is seen over here. Now the question is how to resolve this holiday junction. For that I would like to focus you over here. See, there are two possibilities. I will use laser pointer for this. Okay, just wait for a minute. If I have the other color for this, I would like to use other color. Let me see if I have. I don't want. Yes, I can have the purple color for laser pointer. No, laser pointer. Okay, it is available only in red color. Doesn't make any difference. You can see these two intermediate with two holiday junction. Now there are see two holiday junctions are there. For our convenience we will label it as X and this is the Y. Okay, now see there are two options. This if the junction resolved by having the vertical cut, then we call it as a cutting at one position. Cutting at one. Whereas if the cutting resolve, if this holiday junction get resolved by cutting horizontally, we call it as a cutting at the second side. Okay, so there are two options, either the vertical cut or horizontal cut will take place. Let's see one by one. If the cutting takes place horizontally in if the cutting takes place horizontally in both the junction X as well as at junction Y. Again I'll repeat, if cutting takes place like this way, that is the horizontal cutting at junction X, and like this way the same horizontal cutting at junction Y, then it results in formation of this product which is called as a non-crossover product. Later on I will tell you what is that non-crossover product, but let's see other possibility. Now here this product is formed when the resolution at junction X it takes place vertically. If the cutting takes place vertically at junction X and it takes place horizontally at junction Y, then what will happen? In that case it will result in production of the crossover product. Okay, now what is the difference between crossover and non-crossover products? See, in non-crossover product, if you compare, see these are two chromosomes which are produced by the recombination. Okay, now if you look at the parental genotype, the gray color chromosomes was having capital A, capital B, capital A and the capital B, whereas the blue color was having small a, small b, small a and small b. The same genes are seen in this products and therefore we call it as a non-crossover product. But if you look at these chromosomes, which are formed by the resolution at X sides in a by the vertical cutting, whereas at other side by horizontal cutting, then you will find that here the capital B, I mean the gray color, now it is with the blue color, that is the small a is with capital B and capital A is with small b. So this is nothing but the crossover product, isn't it? So this results in the formation of the crossover product, and this crossover product are it is totally different than either of the parental DNA. So this is known as the recombinant DNA, which is different than the parental DNA. Thus, this recombination, it is, we can call it as a evolution device, because it explore the new combination of sequence. So, what is the benefits of recombination? So that results in formation of a greater variety in the offsprings, because it generates the new combination of allele. See, new why I'm saying a new combination. The previously it was small a, small b, capital B. But if you look at this crossover product, you will find that the capital A is now associated with the small b, and small a is associated with the capital B. Thus, because of recombination, that leads to generation of new combination of alleles.